Daniel, and I'm excited to talk about neural embeddings for amino acid relationships. And a little bit silly of me, I forgot to put on the slide, I'm from the University of Montana. My advisor is Travis Wheeler from the University of Arizona. So the story of NEAR begins with sequence similarity search. Sequence similarity search is exactly what it sounds like. You take a query sequence and you take a collection of target sequences, and your goal is to find, well, similar query target pairs. And this example is an easy example. Sequence similarity search is a problem that is getting more and more difficult, and is getting more difficult because we're getting better and better at sequencing. And so as we get better at sequencing, our sequence databases are growing. So there's this large computational problem where we are in desperate need of methods that are efficient enough to actually work with all of the data we have at our disposal. And also there's this problem where we have so much diverse sequence content. We can now sequence microbial communities, and the proteins that you get out of those microbial communities are incredibly divergent relative to things that we know about. And we need methods that are sensitive so that we can actually understand what's happening inside of these microbial communities. So there's a bunch of different ways that people have thought of trying to improve sequence similarity search. With the advancements and successes of deep learning, people have, of course, thought of applying deep learning to sequence similarity search. And there's sort of a bunch of different ways you can go about this. A fairly common approach is to take a neural network and use that neural network to embed your sequences. So you feed your sequences into the neural network independently, and the neural network transforms those sequences into high-dimensional vector representations. And the idea here is that a smart neural network is going to be able to cluster those representations in their high-dimensional space according to some latent metric of similarity. So in our example, Q and E were the most similar sequences. And so we might expect that Q and E would be very close together in this embedding space. And they are in this cartoon. So there's two really big advantages to this if you can get this idea off the ground. One of the big advantages is that perhaps neural networks can learn subtle patterns of similarity that are meaningful but very subtle and hard for classical methods to recognize. Another really big advantage is that by recasting the problem of sequence similarity search into a problem of vector similarity search, you now have all of the algorithmic advantages of approximate nearest neighbor detection at your disposal. So you maybe have the chance to have a very efficient way to find similar sequences. Now, a lot of people have done this sort of thing, and very common is to use a protein language model to do your embedding. And there's all sorts of papers that have shown protein language models can create these incredibly powerful embeddings that can be used to recognize similarities in evolutionary history or function or structure that is completely missed by other tools, but can be verified to be correct. And the results from using protein language models in this way is really, it's quite amazing. There's also sort of a lack of careful benchmarking to ensure that protein language models are not overly discovering false relationships. So one of the things that we wanted to do is develop a method that can find new things, but also we wanted to be careful that we don't find things that are not real. So we wanted to benchmark false discovery rate. So here's a benchmark that we sort of chased after. The construction of this benchmark is like this. We take 10,000 query sequences and 20,000 target sequences out of UNIREF 90. These are full-length protein sequences between 256 and 1,024 residues in total size. And we align these with p-hammer. And once we do this, we get this set of query target relationships with good e-values. These are query target pairs that have meaningful similarity. They have some biological significance to one another. And the goal of this benchmark is to recover all of these. So 100% recall is what you're aiming for. Now, to measure false discovery rate, we rely on a fairly simple benchmark. And what we do here is we have this sequence we call, or this set of sequences we call D-shuff. And D-shuff is our decoy set of shuffled sequences. These are sequences that we take from our target set, and then we scramble the contents of those sequences. They're shuffled, they're random. These are not biological sequences. These are random non-biological sequences. And our goal is that we should be able to filter out all query decoy pairs. There's nothing in here that is related in our decoy set to our query set, so we should be able to filter all of these out. So, and the construction of this graph, upper left is the best, and if you look at classical algorithms, they perform well. So phammer, sort of unsurprisingly, does very well on this. It's unsurprising because phammer was used to construct the benchmark, although albeit under slightly more sensitive settings than is tested in this figure. MLCSB also does very well. The recall is slightly lower, but the filtration rate is quite high. Now, when we test out some protein language models, we find that they don't do quite as well. So PROT-T5 does the best, MLCSB does the second best, and, or excuse me, not MLCSB, ESM does the second best, and PROT-BERT does the third best. Now, I want to note that it would be easy to look at this and say, well, the filtration rate is actually pretty good on these, right? Well, no, they aren't very good on filtration because we're working with large scale searches. So if you are doing this experiment and you're searching against 20,000 sequences, what you would find with this 50-ish% recall and this 99.98% filtration rate, what you would find is that you're getting about 5 true positives and 5 false positives. And if you were to scale this search up to 100,000 sequences or 1 million or 100 million sequences, suddenly the false positives are going to far drown out your signal. Now, there's a few reasons that this can be. We thought that one reason that the protein language models might have been performing slightly poor in this experiment is due to our usage of sequence representations. So protein language models, they take a sequence and they create these token embeddings that are rich with all sorts of interesting contextual information and captures so much valuable information. But then when you create a sequence embedding explained in the previous talk, you can end up losing a lot of information. And our thought was that by using sequence representations, we are effectively muddying the water, and we lose so much signal that it's just impossible for these protein language models to actually perform well in this experiment. So we thought that instead of using sequence representations, we would use token representations. And so here we're using the representations for individual amino acids, and we perform our search. And I'm not going to describe this method in any depth. Feel free to look at the paper. But we do this, and here are the results we get. Not a big improvement. First off, let me explain. The lines here are representing what would happen if you consider different score thresholds, different similarity thresholds. What would the filtration and recall be for that particular threshold? And what we see is, once again, PROT-T5 does the best, ESM does the second best, and PROT-BERT does the third. None of the methods are improving by all that much. PROT-T5 does have a fairly substantial increase in recall, but only at the cost of filtration. And I want to be careful about the interpretation of these results. The interpretation is not that the protein language models are bad or that the embeddings are bad. There are very good results that show protein language model embeddings are good. They can be used for all sorts of very, very sensitive discoveries. Also, protein language models embeddings are very, very complex, and it encodes a lot of information. It's very easy for that information to not be particularly interpretable. It's very difficult to use protein language model embeddings and interpret the results of embedding similarities with very much rigor. So this is in part good news for sort of little researchers like myself, where I now get to use a model that is not a general purpose model, but I get to develop a model that is specialized for a specific task, and in particular, specialized for sequence similarity. So NEAR is a sequence similarity model. It's built on a, that is something. Okay. So NEAR is a simple, small 1D ResNet. Excellent. So NEAR is a simple 1D ResNet, but we train it with sort of a particular contrastive learning approach that allows it to perform very, very well. There's also a part of NEAR that is fast vector search, and there's a part of NEAR that is using the results of that fast vector search to actually infer sequence similarity. And there's a whole bunch of recent improvements into NEAR that I'm not even going to talk about, but I suppose the point is that NEAR has a lot of methods to it, and I'm not talking about those methods right now. I'm mostly just talking about results. And when we look at the results of NEAR, what we find is comparing it to previous, on this benchmark we developed, we find that NEAR does well. So NEAR is these two lines up here, and it's hard to tell, but it asymptotes on the vertical axis, which is very, very good. It has comparable performance in this benchmark to classical methods. Very, very good filtration rate, very, very good recall, and also I will draw your attention, it is significantly faster than classical approaches and also much, much faster than protein language model approaches. These results are good. We're happy with these results. We've also, like I mentioned, we've done some recent improvements and we have better results now. These are not peer-reviewed results, but in a slightly harder benchmark, we are finding that NEAR is outperforming classical alignment-based tools. And I will also note that NEAR is 10 times faster than MMCX and 100 times faster than pHammer. So that is NEAR. NEAR is fast, it is sensitive. We would like to demonstrate that NEAR is able to find things that other tools miss. That analysis is going to wait for the future. And I think sort of an auxiliary takeaway from NEAR is that although protein language models are really great, there is a lot of room for deep learning research that does not take hundreds of GPUs and huge parameter spaces. And with that, I would like to conclude this talk. Thank you all for listening. Oh, and before I conclude this talk, I do want to mention I just finished my PhD, so if anybody would like to hire me, we should talk. That is it. Thank you very much.
All right, questions?
Yeah, great talk, Daniel. Thanks very much. The speed is really impressive, but I wonder, is there some pre-compute steps that aren't included here for calculating embeddings and making indices and these sorts of things?
The compute times shown in the paper and in the first, let's see, in this benchmark, these compute times include everything, indexing and embedding times. This does not include indexing time. The indexing time for this improved method is sort of complex to calculate. The library that we're using is undergoing some maintenance. So when we publish these results, we'll include that indexing time also.
Great, thanks.
Go ahead.
Hi. You said that you expected that NEAR would be able to predict things or discover things that other tools are not able to. So what sort of things are you thinking of and why?
For NEAR, we are specifically interested in distant homology, distant evolutionary signal. A really big distinguishing point between NEAR and a protein language model-based approach is that NEAR is not trying to understand deep structural or functional information. It's really just looking at that primary sequence and trying to understand the primary sequence as best as it can. And so I suppose to answer your question, the new relationships we would like to uncover with NEAR are entirely distant homologs, remote homologs, these long distance evolutionary relationships.
Yes, number 2.
Hey, Daniel, nice talk again. I've seen it before. And I have a question about interpretability, because you mentioned that the black box, the protein language models are kind of black boxes and that it's really difficult to interpret. I believe it's in the paper, but can you talk a little bit about interpretability and how NEAR works with interpretability? How easy is it to interpret these results?
Well, thank you for the question, Jason. Interpretability is difficult for deep learning models in general. We have results that are not yet published that show that NEAR has some advantages in interpretability. So specifically, using just linear projections of NEAR embeddings, you can squeeze every bit of information out of a NEAR embedding. What we found in our research is that using protein language model embeddings, you actually do need a neural network to squeeze as much information out of those embeddings as possible. And what that means is interpreting the protein language model embeddings is dependent sort of on the amount of compute you throw at it. There's advantages to that in that the embeddings can contain more information, but there's disadvantages in that the embeddings are then less interpretable. A significant advantage to NEAR embeddings is that the linear projection, being able to just linear project the embeddings and get information out of it in that way allows us to build a probabilistic model from NEAR embeddings. And so we're actually able to get a surprisingly good p-value estimate from NEAR embeddings and using NEAR search in our recent improvements. We're very excited about that.
Any more questions? Okay, if not, let's thank Daniel again.
Thank you.